Hello everyone. In this session, mainly we are going to study about the mechanism of transcription of the chapter Nucleic Acid, which comes under the paper Cell Biology and Biotechnology for final year B.Sc Zoology students. So what is transcription then? So we have gone through the replication. So before that we have studied central dogma of molecular biology, that is first replication, transcription and translation. So translation, it is nothing but the process that helps in the synthesis of RNA by using DNA as the template, and transcription is DNA dependent RNA synthesis. So as you can see in this picture, how the RNA polymerase is getting worked with DNA, and later that leads to the formation of RNA. So this is the DNA, and RNA polymerase is an enzyme which helps in the formation of RNA, and that is called as the transcription. So before studying the transcription, you need to know about transcription unit. So how this transcription unit works. So it is nothing but a part of DNA that takes part in the transcription. It has got promoter gene, structural gene, terminator gene. So promoter, it starts the action, and structural gene which helps in the movement of these enzymes and also production of RNA. Terminator, it helps in the stopping the reaction. So these are the three important genes, or you can tell the unit of the transcription unit, that is promoter, structural gene and terminator. So this transcription, it has got two important steps in eukaryote as well as in the prokaryote. So for the transcription, what all are the requirement? So DNA strand should be there, and ribonucleotides, that is ATP, GTP, UTP, CTP, and RNA polymerase, very important enzyme, and divalent metal, that is Mn and Mg. So both, or it can be neither of that, and protein factors. So these all are the requirements for transcription. So it involves three important steps, that is initiation, elongation and termination. So in initiation, you know how RNA polymerase it unwinds, and later the synthesis of the RNA with the help of RNA polymerase enzyme, and later elongation that leads to the formation of RNA strands, and termination stopping the RNA synthesis. So let's see in the detail. So the process of transcription, as you can see in the picture how it is taking place, it starts from the 5' to 3' direction, and the polymerase enzyme movement, it is taking place at the specific region, and there NTPs. So usually there what happens, the synthesis of mRNA on the strand of the DNA, that is the transcription, and usually it takes place in the nucleus of the eukaryotic cell and cytoplasm of the prokaryotic cell. And transcription, it begins with the uncoiling of the DNA strand, and thus that leads to DNA molecule at the specific site, and that is called as promoter sequence or promoter gene. So this RNA polymerase recognizes and binds to promoter sequence. Once the binding of RNA polymerase takes place, later the what happens, the one DNA strand, this act as a template and helps in the synthesis of RNA strand. So that is called as antisense strand or non-coding strands. So the coding strands, and non-coding strands, they are also called as the sense strand. And RNA nucleotides, they are led complementary to antisense strands. So there the RNA polymerase enzyme which will help in the polymerization of the nucleotides. So once the polymerization takes place and synthesis of RNA strand, it takes place always from 5' to 3' direction. So this polymerization or the synthesis of RNA, so during that time RNA polymerase reaches the termination, that is terminator region. So there the RNA synthesis is stopped. So these are the very important steps that comes under the transcription. So when you are differentiating with along with the eukaryotic and prokaryotic, so here you can see the initiation, elongation and termination. So there are two important factors, that is sigma factor and rho factor. Sigma factor which attaches to the promoter and starts the synthesizing, and rho factor during the termination it stops the synthesizing. They are the main component of the RNA polymerase enzyme. But whereas in eukaryote you can see the splicing taking place and polyadenal capping taking place. So there poly A tail formation takes place, and thus the messenger mRNA that is formed. So these are the steps you need to know how the transcription takes place both in prokaryotes and eukaryotes. So this is the difference between prokaryotic and eukaryotic transcription. So as you can see, under prokaryotic transcription it occurs in cytoplasm of the cell, whereas in eukaryotic it occurs in nucleus, and the transcription and translation happens simultaneously in prokaryotes, but whereas in eukaryotic first transcription and later translation takes place. mRNA is transcribed directly in prokaryotic cells, but whereas in eukaryotic cells initially pre-mRNA, that is precursor, so they are formed and later they yield matured mRNA. And RNA polymerase consists of 5 subunits, but whereas in eukaryotic it has got more than 12 to 15 subunits, that is 10 to 17. And holoenzyme, that is they act as RNA polymerase plus sigma factor, but here promoter gene recognizes cannot carried out by the RNA polymerase alone. So here there will be an complete enzyme, but here in the prokaryotic there will be a holoenzyme. So in this session mainly we have studied about the transcription, very important for your examination point of view. They can ask about what do you mean by transcription for 1 mark, what is transcription unit, and what are the enzymes and the mechanism that takes place between prokaryotic cells and eukaryotic cells, or the difference between prokaryotic and eukaryotic transcription. So that's all about this session. Thank you.